Okay, so we said that last week we begin with so this is a deconstructionist view. So back in the day, they used to consider anatomy at the gross level. So we have only gross anatomy. Then we arrive to histology, which is so what's the difference between gross anatomy and histology? So gross anatomy can be seen with the naked eye. You can see the anatomical structures. You don't need a microscope to see the heart. You don't need a microscope to see the lungs. You don't need a microscope to see the muscles. But you do need a microscope to see the cells, and that is histology. So deconstructionist says that we reverse the pyramid. So we are not just so anatomy is not only happening at the level of big up here. Organism. That is the biggest organism. Organism is made up from organ systems, and the organ systems are made up from organs. So these are the organs, and then the organs are made up from tissues, from tissues, from tissues, and then the tissues are made up from cells. And then the cells are made up from atoms, made up from molecules, and made up from proteins and enzymes and all of that stuff inside the cell. So in the past, the pyramid was inverted. So it was constructionist. We used to look at self as a whole, but now we're looking at the parts that when we add them together, the sum of these parts will give us the whole. You need to know that. You need to know this order. So you should be able to tell me that how do we go from organism in a deconstructionist view? You say we have an organism, and the organism is made up from organ systems, and the organ systems are made up from organs, and the organs are made up from tissues, and the tissues are made up from cells, and cells are made up from molecules. So the cells and organelles, don't forget that the cells are made up also from organelles. So the cell has endoplasmic reticulum that is responsible for the packaging, and it has ribosomes attached to it. There are freely floating ribosomes in the cytoplasm. The cytoplasm is another organelle. Then you have the nucleus. Inside the nucleus, you have the DNA. And in the DNA, we have our central dogma of biology. That one you have to know. What is the central dogma of biology? We mentioned that last week. Yes, excellent. We go from DNA to mRNA through transcription. And we go from mRNA into proteins through translation. And then the proteins, we have two types. We have two types of proteins. One that is known as structural proteins, excellent. And the other one is known as functional protein. Structural proteins and functional proteins are happening inside the cell. My cell cannot survive. So the cell is the basic unit of life. We are alive because the cell is alive. We are dead because the cell is dead. We are healthy because cells are healthy. And we are sick because the cells are sick. So we don't just look at the cell, we look at the cell's building block. And there are very important proteins and enzymes. These proteins and enzymes, they regulate the cell, the process of cell division. How is the cell now able to divide? Because of proteins. Proteins tells the cell there was damage, and the immune system macrophages came and they removed all of the bacteria. The infectious microbes are now gone. The parasites are now gone. No more fungi, no more bacteria. So we removed all the damaged tissue. So now we need to make new healthy tissue in place of the old one that we removed. Or do we just remain there with no cells? So now the cells have to Now go in the cell cycle and divide. So when the cell cycle divides, who regulates that? Enzymes. So enzymes are the ones who regulate the cell cycle. Not only the cell cycle, there are many other enzymes doing many different processes, like the metabolism inside the cell. We said last week we have oxidative phosphorylation. We have aerobic respiration. When we metabolize carbohydrates, we get ATP. Who's doing that? Proteins, enzymes. Who is metabolizing for me the fats into fatty acids that will give me ATP? Enzymes. Who is able to metabolize for me proteins to get amino acids and get ATP, energy out of them? Enzymes, enzymes are proteins. So the cell itself is not functioning. You can have a house, but if there's no AC in the house, there's no stove in the kitchen, the bathroom is not working, do you really have a house? So the same thing, just because you have a cell doesn't mean you don't need the organelles. Sometimes one protein damage mess up the whole cell. Now the cell has to die. So don't forget that. And you gotta know this organ. So now you know that the atoms and the molecules give me the cell, which is the basic unit. Then many cells together give me tissue. Then many tissues give me a. So give me an example of an organ system now. What's an organ system? So yeah, digestive system, but how is it an organ system? So the digestive system has the stomach. The tissue of the stomach looks different than the tissue of the intestine, different from the tissue of the esophagus, different than the tissue of the cells in your buccal oral cavity, different from the tissues in the anal canal. So these are different tissues to give me the organ system. In our skin, we have the skin of the epidermis and the dermis. These are the epithelial cells of our skin. But do they not have hair cells, keratin? Do they not have nerves? Do they not have blood vessels? So that these are different organs working together to sustain life. This is golden. It makes no sense that a student is in anatomy and physiology and they cannot arrange that. They put cells after tissue and they don't know who comes first, who's the more basic, who does the atom come before. You have to know this one. Okay? Then that's the picture there. So that shows you in the picture there. Is there like a here some sort of there's no projector, right? Yeah. Then the word escaped my mind. Projector. It's gone. On my dictionary. I was saying that there's no she said projector. Huh? You know what you meant. Yeah, okay, good. You see that's we're gonna get into the nervous system. That's the beauty of the central nervous system, the brain cells. They are able to internalize and integrate information. You can integrate emotion. Somebody doesn't have to say anything. You look at their face and say, "Oh my God, he is so angry." Just by looking at their face, or you can say, "Oh my God, he's so happy." Oh, she's so sad. So that's the emotion. So I was trying to say the thing, it's gone, but you still understand it. So that's normal. That's intelligence. Okay. So and then many organs. We're not done here. We're not done. That's not the highest thing. That's the highest thing we're gonna know for homeostasis. Then there is one level up there. We don't put it there, but you have to know we're gonna deal with it later in the course when we move on to anatomy and physiology too. Who's the up there? Who's above the organism? Excellent. Who said that? Very good, Jennifer. Excellent. The population. So always, when you want to know what's happening upward, double the ones who are below. Many atoms and molecules give me many cells. Many cells give me many tissues. Many tissues give me many organs. Many organs give me many organ systems. Many organ systems give me an organism. Many organisms give me a population. So now do we see this deconstructionist view that we have deconstructed a human being from top? He was an organism. We divided him into, you see the organ system. then the organ, then the tissue, then the cells, then the organelles, then the micromolecules, then the molecules, then the atoms. So you see now, that's a key point. That's why I love the figures and the figures and the illustrations, because they are the ones that have the key concept. Maybe you look at the figure, you understand it really well. But if you need more information, I'm not gonna hide the knowledge from you. I think that's unethical. If the PowerPoint is 170, I don't want to hide from you. I give you only 50 and keep the rest to myself. I want to put the whole thing there. Maybe you take it for life. You go to the next course, 10 years from now, you still have, you want to go back to it, to open it like a library. So that's your personal library, but you're not obviously responsible to know everything. So now let's move on to that. So we talked about all of this. You see reductionism. So we used to have holism, and these are emergent properties that occur as we ascend the levels of organization, and these cannot be predicted from properties of individual parts alone. So humans are more than the sum of their parts. So if we go from big to small, that's deconstructionist. And if we go to small to big, what is that called now? Constructionist. Very nice. Who said that? Constructionist. Nice. But the word that they use is over there in the slides. See, if you see all the way in the back, you can see it. I like the word. I'm going to put it. Constructionist. Constructionist. I like it. But they have another word. What is it? You want to look in the slide? I'm sorry, in the back, you told us deconstructionist. That's nice. I like it. I put it. But I want you to give me the exact word they have in the slide. Holism. Holism. So deconstructionist and reductionist are the same. So we reduce. Reductionist and deconstructionist. You know, we reduce. When you deconstruct, you reduce, right? Are we following? Yeah. That deconstructionist and reductionist are the same. In different books, they tomato. And here, constructionist, like your friend said, and also holism, holistically, holism, that's when we go upward. Okay, so you need to know the names of the views and how to arrange them. So if, for example, I can ask you a short answer and say, can you please assemble for me in the deconstructionist and the reductionist view, or from organisms to the atoms? Then you put organism first, followed by organ system, organs, tissues, cells, atoms. Or I can tell you the opposite. Right, for me, the holism or the constructionist view, then we go from small to big. So a deconstructionist, we go from big and we break down, we deconstruct from big to small. While here is the opposite. We go from small and then we build big, 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 big. Okay? Okay, so what does this tell us? That tells us that in anatomy and physiology, we are going to be studying the structure and function. What did we say about the structure and function? What did we say about the structure and function? I'm sorry? What do we say about the structure and function? Remember, I told you, if we have steel, we can get a knife and we can get a hammer. In steel, we have a knife and a hammer. But the knife can cut and the hammer can hammer down. So that shows you our function is from our structure. If the knife wasn't structurally sharp, if the hammer wasn't structurally dull, it cannot do its job. So we study the structure so we can understand the function. We study the anatomy so we can understand the physiology. Okay? Then that tells you the anatomy and physiology that we are going to be studying. The anatomy and physiology that we're going to be studying, is it for every single human being out there identical? No, it's for like 80% of the people. So what does this graph show you? It shows you that these are the normal kidneys. They're supposed to be looking like that. So I think if you guys face that way so you can see the mouse. So you can see my mouse pointing. Okay? Yeah, please turn so you can see the mouse turning. Thank you so much. You see, this is the normal kidney. That is how most of the people's kidneys look like. That is how most people's kidneys. Okay. Can you guys see? That's how the kidneys of most people, the kidneys of most people look like. But some people have kidneys in the pelvis, kidneys in the pelvis. Some people have horseshoe kidney, horseshoe kidney. So these are two aberrant, gross, pathological malformations from my normal kidney. So I can ask you on the exam, give me two examples of abnormal kidney anatomy. You say pelvic kidney, somebody has a kidney in the pelvis, or horseshoe kidney. Their kidneys are connected like a horseshoe. Then another example would be that's the normal aorta, looks like that. But it doesn't mean every single aorta out there looks like this. So some of them, they have, you see, their branches are different. So another example of variations in human anatomy of the kidney and the major arteries are your aorta. That it may be in some people looks different than in the normal. So these two examples, how the kidney may look and how the aorta may look. Next illustration. This one, oh, this one is golden. This one is very high yield concept. You knowing the steps of homeostasis, we're going to be doing this every single day when we come in. You knowing the 8 steps of homeostasis, we said that for homeostasis, that is our green range. The homeostasis is our green range. So the homeostasis is green. Homeostasis, you guys see, homeostasis is, green is not working. Get another green. Okay, so green for me, that is homeostasis. Green for me, that is homeostasis. 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 Also known as U, EU. That's a prefix. EU is a prefix. You guys see the prefix? Okay. And below my green range, I will always have, okay, so if I'm sorry, if I'm illustrating in the board, look here so you can see. If I'm pointing to the PowerPoint, look there so you can see. Okay, so now we're talking about this. That's the homeostatic green range. Below it is my yellow. You know, the traffic light has yellow, green, and red. So this yellow region is my adaptation. My adaptation part. My adaptation part. Okay, and then if you cross the line, after yellow, you go deeper than yellow, then that is known as the line of damage. We said it's the line of damage beyond salvage. This is the line. You cross it, it's going to be an irreversible damage. That's damage beyond salvage. Okay? And that's homeostasis. And the yellow is my adaptation. I'm trying to adapt. Adaptation. 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 The yellow is my adaptation. Okay, so if I go upward, there's, see, an upward deviation from homeostasis. If I go up, that's an increase. That's an increase. That's dangerous. When you go up, that's plus, plus, plus. That's dangerous. That's hyper, hyper. The prefix is hyper, hyper something. And if we drop below homeostasis, that is known as hypo. Excellent. Hypo, hypo. That is known as hypo. 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 That is known as minus. We go down, minus, 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 and that is known as the prefix hypo. Hypo something. I have mentioned for you already the steps that we need to take to repair homeostasis. Remember? So we said, how many steps do we have to repair homeostasis? 6, 7, 8, the auction. 8, 9, 10. Okay, number 1, we have a signal. So we develop them as we go along. The first step is we have a signal. A signal. A signal is also called a stimulus. A stimulus. A signal or a stimulus. So the signal, I should have drawn it in red, because the signal, the stimulus will always be one of these two guys. Either there's some hyper change or there is some hypo change. Are we following? Either hyper change or hypo change. Then we move from signal to the next step. That signal must be detected. The signal must be detected, number 2, by a sensor. By a sensor. So if you have a signal, somebody got to detect the signal. If you have a stimulus, somebody got to detect the stimulus, or a detector. Detector. And the detector will be either a chemoreceptor to detect some changes, hyper or hypo change in chemicals. Chemoreceptor. Number 2, it could be an osmoreceptor. Number 3, it could be thermoreceptor. Number 4, it could be a stretch or pressure receptor. Stretch or pressure receptor. And number 5 is nociceptor. Yes.
Is the stretch pressure receptor same as the mechanoreceptor?
Yeah, excellent. Stretch, pressure, mechanoreceptor. Mechanoreceptor. Then we move to step number 3. What is step number 3? Now, we have to have an input. An input sensory signal. An input sensory signal. So the input sensory signal, that's step number 3. And the input sensory, are you guys following? The input sensory signal, number 4, will now send the information to an integrator computer control center. So the integrator is my computer. The integrator is my computer. and the computer will integrate for me the input and generate an output. So like a computer, this very important integrating center, known as the control center, or integrator, or for integration, and that would be step number 5. That's step number 5. Then we're going to move on to step number 6. Step number 6 is now we're going to have number 6. We're going to have, if we had an input come in, now we're going to have a, if we had an input coming in, now we have a output. And if the input was sensory, the output is, if the input is sensory, the output is motor. It's going to be motor. The input is sensory and the output is motor. That's step number 6. Then the output is going to go to step number 7. And step number 7 is the effector. The effector organ, always an organ, is going to be the effector. The effector organ. Then the effector organ is going to restore for us step number 8, and that is going to be in all green. That's what we want. We now going to restore step number 8. And step number 8 is, what is step number 8? Homeostasis. Step number 8 is homeostasis. Step number 8 is homeostasis. So homeostasis. Then after I restore my homeostasis, the last step is this step number 1, 2, 3. You see, I put 4 and 5 at the same step. So 4 is my integrator. I made a mistake. So the 4 is the integrator. So 4 is my integrator. 4 is my integrator.
It's not supposed to say something about effector.
Right here. 4 and 5 is my output. Okay. And 6 is my effector. And 7 is my green homeostasis. 7 is my homeostasis. And 8 is I gotta stop. We gotta stop and terminate the response. Stop and terminate the response. We're gonna stop and termination. Stop and termination. Stop and termination. Stop and termination of the response. Stop and termination of the response. So these are the steps that we're going to be taking to repair our homeostasis. Now, let's go through these steps for our variable. So now let's talk about temperature. When we are so hot, what happens? When we are so hot, is that euthermia? So let's begin with the first variable that we want to discuss. Begin with temperature. Temperature is known as thermia. So our euthermia is 37 degrees Celsius. That's euthermia. If you are at euthermia, will there be any sensation? Will there be any sensor signal? No. Let's say now you went up to 47 degrees Celsius. So that's an example of hyper what? Hyperthermia. Very good. That's an example of hyperthermia. This hyperthermia is going to be a signal and a stimulus that there is hyperthermia. The hyperthermia is going to be detected by this hyperthermia signal. This hyperthermia stimulus is going to be detected by a chemoreceptor or a osmoreceptor or a thermoreceptor or a stretch pressure mechanoreceptor or a nociceptor. Which of the five is going to detect my temperature? Thermoreceptor. Excellent. Then the thermoreceptor is going to send an input to the computer. Input that, oh, there is a lot of increase in temperature. It's going to send that there is a lot of increase in temperature. There's plus, there's hyperthermia. So the control system is going to integrate the input and it's going to generate an input that is the opposite or in the same direction. It's going to be the opposite. That's why we say negative feedback. Imagine you're so hot and the input says to the computer, you're so hot, and then the computer says, make me more hot again. So now we're going to go from 47 to 57. That would be an example of positive feedback. We don't want positive feedback. So we repair homeostasis with negative feedback. So you see, negative feedback means the input sensor signal will always be the opposite of the output signal. You see, the input signal is always going to be the opposite of the output signal. So here we are so hot and we are suffering from hyperthermia. The control center will be your hypothalamus. We're going to get to that, don't worry. Will be the hypothalamus. And the hypothalamus has multiple nuclei inside it. One is called the thermal nuclei. You know, in the police, they have a division for robbery, a division for homicide, and a division for the different types of fraud. So the same thing for a hypothalamus. It has a nucleus for changes in temperature. So the thermal nucleus is going to generate a response that now let's bring down the temperature. So now we're going to send a response to bring down the temperature minus hypo. So you see how they are reversed? The input is hyper, the output must be hypo. Otherwise, I'm going to go even worse. I'm going to go even hotter. Is this point clear?
Yeah.
Okay. Then the output is going to be, let's warm things up. How? We're going to target the effector organ. We said for thermia, we have two effector organs. The first one is the blood vessels. So the blood vessels now are going to start dilating. And when they dilate, they accommodate more amount of blood that is flowing near our skin. And we said from our diffusion lab, we said in the diffusion lab that if things are so hot, then they move from an area of a higher heat, like here, 45 degrees Celsius, into the cold air that is 25 degrees Celsius, and heat is going to leave your body, leave your skin, leave your blood. The hyperthermia is going to leave your blood into air, and then as the heat is leaving, what happens to your temperature? Is going to go down from 45 to 44, from 47 to 46, 46 to 45, 44, 43, 42, 41, 40, 39, 37. Then we stop here. Then when we get to homeostasis number 7, then we stop. Because if you keep on dilating the blood vessels even more, we're going to keep going more down, 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 down. Make sense? Okay, now let us also look at the second effector. The second effector would be the sweat gland. So the sweat glands are going to secrete sweat. And the sweat has a lot of water. So the sweat now is also going to help cooling you. Then when you cool, Then when you cool the sweat, and then when you cool down, you reach homeostasis, then you stop. You terminate the response. Is that clear? Any questions? Does anybody have any additional questions? Does anybody? Yes.
Not a question. Maybe we can
Lower the AC. So I don't know, and I can't touch that. So no, they don't give us that. If I touch it, then a problem happens, then we're gonna be in trouble. So like that? All right. Okay. Okay, guys. So now we have repaired hyperthermia. What about the opposite now? We're no longer dealing with hyperthermia. We're now going to deal with hyper something else. So let us now repair that. Let's repair something else. Let's now deal with when it's too cold. When it's too cold, the signal is going to be hypothermia. It's too cold. That's going to be hypothermia. We have went down. So now we're no longer 37. We're now 27. So what happens is a signal or stimulus would detect that there is a drop in temperature. Then a sensor or a detector is going to detect for me that there is a drop in temperature, thermoreceptor. Then there's going to be an input sensory signal that's going to tell me that there is a drop in temperature, there is hypothermia, there's a drop in temperature, there is hypothermia, and this hypothermia is going to be detected by the thermal nucleus of my hypothalamus, and then the control center is going to integrate for me that I need a response. If my input was hypo, what should my output be? What should my output be if my input is hypo? Hyper. Should be the opposite. What do we call that? If the input and the output are opposite to each other. If the input are low and the output is high. If the input is minus, the output is plus. If the input is hypo, the output is hyper. What is that an example of? Negative feedback. Excellent. So now we're going to send an output motor response to tell me that I need to do negative feedback increase. And then when we increase, we're going to first target the blood vessels. When we are hot, we dilate to increase the diffusion. And when we are cold, what do? We shrink, but we don't say shrink. We say vasoconstriction to prevent the loss of heat. So because now there's not enough blood flowing, so not a lot of heat is leaving us. We are constricting the heat in our body. Because if we leave the little bit of heat that we already have, we're going to get even colder. So now we're going to conserve the heat. That's blood vessels. So now we're going to have vasoconstriction. And when we're so hot, we have vaso. Yeah, when we're so cold, we have vaso. Excellent. Then the other effector organ is going to be my skeletal muscles. And my skeletal muscles are going to receive electrical energy, electrical stimulation from my hypothalamus to do an action potential. And then the action potential is going to cause shivering of my skeletal muscles. And then the shivering is going to convert for me ATP. Then I'm going to use the ATP fuel like gasoline, like Bunsen burner to warm myself up. And when I warm myself up, what happens? I achieve homeostasis. So now I warm myself up. I go 27, 28, 29, 30, 31, 32, 33, 34, 35, 36. I'm at 37. I'm at homeostasis, green now. Then I got to do stop. Stop what? I'm sorry? Stop what? Terminate what?
Elevation.
I'm sorry? Terminate? Yeah, so we stop the effector. We stop this output, this sending information to vasoconstrict. We got to stop vasoconstricting anymore. We're done. And we got to stop shivering. We got to stop shivering thermogenesis. We got to stop the skeletal muscles from shivering. And because we have already arrived to the destination. With the GPS, when you get home, you don't run through the garage. You stop there because you have achieved homeostasis. So now, now you can look there. Now you can look there. Okay, so the negative feedback in thermal regulation, that tells you actually what happens in your room. So in the room temperature falls to 19 falls. The key word here is falls. So what kind of signal is that? What kind of stimulus is that? Thermal, yes. What kind of change? Is it hyper or hypo? Hypo what? Hypothermia. Very good. What does this arrow indicate for me? Yeah, input sensory signal. And the thermostat will activate the furnace. What does the thermostat represent for me? Integrator, control center. Excellent. Then the integrated control center send what kind of signal out? Increase the temperature. So it received fall in temperature and it sent out increase in temperature. What kind of feedback is that? Negative feedback. Excellent. Then the furnace is going to represent my effector. Excellent. And the effector is going to send out heat. Then the room temperature rise from 19 to 20. And now when we hit the 20, we don't want to go even hot. We are 100 degrees Celsius. We burn. We got to stop. Then the thermostat shuts off the furnace. Then the room cools down. Then we achieve our homeostasis. Is that clear? Now that's what happens in the room. This is what happens in your blood, what we just went through. Thermoregulation, when we regulate temperature. So the middle line here in black, that's the set point. That's euthermia. 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 So euthermia means the temperature went from 37 degrees Celsius to 37.5. Is that hyper or hypo? Is that an increase or decrease? So that's 37.5 degrees Celsius. That's the increase. The temperature went up. When the temperature went up, how did I repair that? I repaired it with vasodilation and sweating. Then when I vasodilated and when I sweat, what happened to the temperature that was going up to here? Now what happened to it? It started going down until I have now a set point. What about the opposite? If I had 37 degrees Celsius, now I go down to 36.5. What is that an example of? Hypothermia. In hypothermia, then I do vasoconstriction, and then I do shivering, and then I return back to the set point. Is that clear? Same thing with now we're dealing with another variable. Now let's look here, please. Let's look here, please. Now we're dealing with no more thermia. We're now no longer dealing with thermia. We're dealing with something else. So when you wake up in the morning, we said, when you asleep, then you get up in the morning, you stand up, the blood pools and falls from your brain and your head and your neck starts to drop down. So what is that an example of? Hypertension or hypotension or eutension? I'm sorry? Raise your hand if you think that's an example of hypertension. Raise your hand if you think that's an example of hypotension. Perfect, because the blood pressure went down. So when blood pressure went down, why is that a problem? That means there's not enough blood getting to the brain. Means not enough oxygen, because if there's no blood, enough blood going to the brain, not enough oxygen. If there is not enough oxygen, there is not enough. If there is not enough oxygen, there is no enough ATP. If there's no enough oxygen, no enough ATP, no enough removal of metabolic waste that is building up. Now your brain is gonna have brain tissue is gonna die. You're gonna have a stroke. So now we're gonna repair that. So we had eutension, but when you wake up, it fell down. That's hypotension. So what is the signal, the stimulus? What is the signal and the stimulus when the blood pressure falls down when you wake up in the morning? Hypotension. Hypotension, minus, minus. Who is the sensor and the detector? Is it a chemoreceptor? No. Is it an osmoreceptor? No. Is it a thermoreceptor? No. Is it a stretch pressure mechanoreceptor? Definitely. So how does it work? We said around the blood vessels, there are stretch receptors. Stretch receptors. They sense the stretch. If the blood pressure falls, how do they sense? Because the blood movement is so turbulent. It is so turbulent, hitting the sides of the blood pressure of the blood vessel wall. So when it hit it too hard, it will detect hypertension. When it hit it too low, it's going to detect hypotension. Excellent, 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 excellent. So that's how hypotension is detected. Then there's an input sensory signal. There's an input sensory signal. It's going to say that we have hypotension. We just woke up. Blood is pooling. Blood is falling. Blood is pooling. Blood is falling. And that is not going to be detected by the hypothalamus. I told you already that we don't have the same player on every team. Just because you are a quarterback in the New York Giants doesn't mean you will also be a quarterback in the Jets and in the other teams as well. So now the integrator is in the brainstem, not brain stem. It is not in the hypothalamus, in the cardiac center of the brainstem. Then it's going to send out, that's going to be my cardiac center. That's my control center, the integrator. Then it's going to send out an output that is opposite or in the same direction. Opposite. That's an example of negative feedback. Then the output is going to tell who's going to be my effector now to bring down to raise the pressure. Because I had hypo, now I need to have hyper. Who's going to respond now? The heart. So the heart rate, the heart is going to be my effector. And what's going to be the effect? The heart rate is going to be going up. The cardiac output is going to be going up. The contractility is going to go up. I'm going to contract some more. Then I will arrive at my homeostasis. Are we clear? Then I get my homeostasis. Do I keep going now with more heart rate going up, or do I stay here? So I stop. I terminate. I terminate what? I terminate the integrator from sending any more output to the effector to raise the heart rate. Then we stop. Is that clear? This is so important. You got to see this everywhere. From now until the last chapter before reproduction next semester, whether you're going to take it with me or with some other professor. Keep this in mind. Use this template to help you understand how we restore homeostasis. This is the template for all variables. No matter if I'm dealing with the cardiovascular system, the endocrine system, the integumentary system, we're going to be using the same template. So just put them in the right place, then you're going to be able to understand. So now let's look there, please. So the homeostatic compensation for postural change in blood pressure. So the person rises from bed and the blood drains from the upper body, creating homeostatic imbalance. And there are baroreceptors. So now the baroreceptors are a type of a pressure receptor. There are a type of stretch receptor. They're all receptors. So that's why I have a separate name for it, because there are also stretch receptors in the cervix. When the baby is pushing, that's a stretch receptor. When the baby is suckling the breast milk, that's also an example of stretch receptor. So I got to give a different name to the stretch receptor in my heart. So when you hear baroreceptor, you should always remember where is that receptor? the heart. Okay, and then the effector is going to be the heartbeat accelerates in response to the cardiac center. Okay, now positive feedback is the opposite. In positive feedback, what's going to happen between the input and the output? Excellent. So here, number one, the head of the baby push against the cervix. You see that's the head of the baby pushing against the cervix. When the baby pushes against the cervix, who's gonna detect that movement? Is it a stretch receptor or a thermoreceptor or a osmoreceptor or a thermoreceptor or a nociceptor or a chemoreceptor? That's a stretch receptor. So it's gonna send an input signal to the brain, and the brain is gonna generate an output signal that stimulates the pituitary gland to secrete oxytocin. Then oxytocin will stimulate the uterine to contract and push the baby out. So here the baby pushes, there is hyper pushing, then in the end there is even more pushing of the baby. What is that an example of? Positive feedback. So you need to be able to tell me an example of a negative feedback and an example of a positive feedback. The same thing that's happening here in birth is also happening in breastfeeding. So in birth, the more baby pushes, so in birth, the more the baby push, the more it gets pushed out with oxytocin. So oxytocin stimulates the contraction of the smooth muscles in the uterine. And also when the baby comes out, you give the baby the milk, the baby won't suckle. Oxytocin, again, this time doesn't stimulate the cervix, doesn't stimulate the uterine, it stimulates the milk duct, the smooth muscles in the milk duct to eject more milk. So in one instance, the baby pushes, it gets pushed out even more. In the other instance, the baby suckles milk, it gets even more milk. Are we clear? Okay, the second thing, what's another example of positive feedback? Like the platelet. I said that when we are injured and blood starts leaking out, that's a big problem because now we're going to go from euvolemia, the volume of the blood being the same. We're going to have less blood because blood is leaking out. We are injured. It's hemorrhage. So when the blood gets out, now we're going to have hypovolemia. We're going to have now hypovolemia. Now we're going to have hypovolemia. We don't want that. So that's why we have platelets. So how does the positive feedback happen? It happens with one platelet. When one platelet stick, number two will come sticking, number three will come sticking. So the more platelets stick, even more platelets will start sticking. That's an example of positive feedback. We talked about gradients. So there's a downhill gradient and there's an uphill gradient. And we said that downhill gradient happens when sodium moves from outside the cell to the inside of the cell. But an uphill gradient is when you take where there's already a little bit inside, you push it against where the majority is. And who does that? The sodium potassium ATPase. And then we have different flow gradients from high pressure, where the pump has the heart has the highest pressure and it sends it to where the area of there's low blood pressure, and then you have dietary glucose, and that is when you have high concentration. So this is a pressure gradient from area of high pressure to an area of lower pressure. The blood will start flowing. Also air, it moves from the oxygen moves from air where there's high pressure. When I'm inhaling into my lung where there is less pressure, then into the tissue where there is the least pressure. Then here, glucose moves from an area of higher concentration to an area of a lower concentration. Then here we have electrochemical gradient, where you have positive charges, that's electrical, and you have also sodium. Sodium is a atom, and it is charged atom. So that's electrochemical. It's a chemical and it has a charge. That's why we call it electrochemical gradient. In here, you have minus, 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 minus. It is so much inside, a chemical that's so much inside, and it also has a charge, so it flows out. Then the warm blood is so warm, and the heat gets out through the skin, And when the heat gets out through the skin, it goes from an area of a high thermal heat to an area of a colder region. You understand? That clear? Okay. I'm not going to ask you about that. Are we done? Yes. Yay. Now we have covered all the key concepts. Good job. Any questions? Anybody have a question? Okay, now we are done with chapter 1. We're going to see it in the exam. So now let's go to chapter 2. Let us now go to chapter 2. Chapter 2 is very important because it is now telling us about the chemistry. We have the chemistry of life. So we already established that in the deconstructionist pathway, what happens is in the deconstructionist pathway, what happens is in the deconstructionist pathway, what happens is we break down what's big to its smaller parts. So now if we want to understand how these organelles are working, there are many chemistry concepts we need to have in mind, like oxidation, reduction. That's very important. The difference between oxidation and reduction. Okay, so biochemistry is the chemistry of life, and biophysics is the physics of life. So today we're going to be studying the macromolecules. I mean, it doesn't make any sense that a student in anatomy and physiology or a nurse does not know the structure of an amino acid. What does it look like? I'm not going to ask you on the exam to draw for me the structure of aspartic acid. That doesn't make sense. But you should know the structure, the general structure of what an amino acid looks like. Some students don't know what a fatty acid looks like. Some students don't know what a nucleotide looks like. You show them a structure. What is this? I'm not telling you which of the 4 nucleotides. Is it adenine, guanine, cytosine, thymine? We're not asking that. We're asking what is it? Is it an amino acid? Is it a sugar? Is it a fat, lipid? Is it a protein? And some students don't know. So that's a big problem. So you have to know your macromolecule. What do they look like? You have to know what a fat looks like. You have to know what a protein looks like. You have to know what a sugar looks like. And you have to know what an amino acid looks like. That is key for us to understand. So the 4 that I mentioned, the carbohydrates, the fats, the proteins, and the nucleic acids, these are macromolecules. Excellent. Okay, so we begin with the element. So a chemical element is the simplest form of matter, and it must have unique chemical properties. So each is identified by an atomic number. So now let's establish the difference between an atomic number and atomic mass. Let me pull up my periodic table. Let's go to periodic table. Periodic table. You want to sell me a periodic table? So this periodic table let me get a better periodic table. Oh, there we go. So this number 17. So this number, look, 1, 3, 4, 5, 6, 7, 8, 9, 10. Why are they putting them in this order? Why don't you take B, make it number 2, and HE, make it number 5? Why are they in this exact order? Because that is their atomic number. So Right here, it's showing you. It's showing you that 17 is the atomic number, while 35.45, that's the atomic mass. So now let's describe the difference. What is the difference between the atomic number and the atomic mass? So you have to understand that in the nucleus of all the atoms, I have protons that are positive, plus protons that are positive, plus, and I have neutrons that have no charge, 0. And then I have electrons spinning around the nucleus, where the electrons that are the farthest away, are known as the valence electron. Electrons that are the farthest away are known as valence electrons. Valence electrons. These valence electrons are the farthest away. They are the very farthest away. And these are the electrons that engage in chemical reactions, not the electrons that are spinning very close. Electrons are also negatively charged. Electrons are also negatively charged, while protons are also positively charged. So when an atom is neutral, it means it's not positive, it's not negative. That makes it neutral, a neutral atom. Okay, this neutral atom, it has protons, neutrons, and remember that the protons are positive, neutrons have no charge. That's why they call them neutrons, because they are neutral. And then you have electrons that are negatively charged. Okay. So the atomic number is, what is the atomic number? The atomic number is the number of protons. That's the same thing. The population in the school is the number of students. They are the same thing. They refer to the same thing. So the atomic, if I tell you now the atomic number for hydrogen is 1, what does that also tell you? Yeah, it has only 1 proton. Very good. If the atomic number for lithium is 3, what does that also tell you? It has 3 protons. If the atomic number for boron is 5, that means it also has 5 protons. Okay. Very, very, very good. Now, what we need to understand besides the atomic number, we need to understand the atomic mass. What is the atomic mass? What is the atomic mass? The atomic mass is the total number of protons plus neutrons. Excellent. So if I tell you the mass number, the mass number is the number of my protons to the neutrons. And that now leads me to the isotope idea. This is a very important concept. So the isotope, what's an isotope? Isotopes are different variations of the neutrons. So isotopes are different variations of the same atom. So I have hydrogen, and beside hydrogen, I have also deuterium, and beside deuterium, I have tritium. Okay, so they are different variations of the same atom. Different variations of the same atom. Isotopes are different variations of the same atom. So that means they must all have the same what? So inside their nucleus, they must all have the same number of, if they are going to be isotopes, and I'm telling you already that hydrogen is an isotope, and so hydrogen, deuterium, and tritium are all isotopes. So that means if they are all isotopes, it means they all must have what? The same number of protons. Now, what is the number of hydrogen? What is the atomic number of hydrogen? How many protons does hydrogen have? So that means hydrogen must have one proton. How many protons must deuterium have? One proton. How many protons must tritium have? One. Very good. Then, how many? So they all must have the same number of protons. If the number of proton increase, you're going to change to a different atom. So imagine deuterium had 2 protons. Now its atomic number is, now who's number 2 in the periodic table? Look, that's helium. So now I'm no longer hydrogen. So I can never touch the number of the protons if I want to be an isotope. So they must all have the same number of protons. Then if they have the same number of protons, they have to have something different. That's why they're isotopes. They cannot have the same number of protons and the same number of neutrons. So hydrogen has 0 neutrons. 0 neutrons. While deuterium has 1 neutron. And tritium has 1 neutron. Do you guys get it? Do you guys get it? Oh, I'm sorry, 2 neutrons. 2 neutrons. So now the atomic number for hydrogen is 1 because it has 1 proton. The atomic number for deuterium is, the atomic number is the number of protons. So hydrogen atomic number is 1. The atomic number of deuterium is 1. Very good. If it was 2, then that means it's helium. It's no longer hydrogen. Then tritium, the atomic number for tritium is 1. Then we come to the concept of the atomic mass, which is the number of protons plus the number of neutrons. So now I add 1 proton plus 0 neutrons. What's the atomic mass for hydrogen? It's 1. So it's very interesting that hydrogen has the same number of protons and the same number of it has the same number, it has the same atomic number and the same mass number. So its atomic number is 1, its mass number is 1 plus 0, it's still 1. So it has the same atomic number and the same mass number. Now, deuterium has an atomic number of 1. It has a mass number of 2. Very good. Tritium has an atomic number of 1. And it has a mass number of, somebody said 3 for atomic number. And it has a mass number of 3. So you must know that one. You must know the difference between the atomic number and the mass number. That's what we've been talking about. If we look here, for example, we have our carbon atom. This carbon atom has these electrons spinning around it. Do you see the electrons around the atom? It tells you that red is a proton. Proton is positively charged, P plus. Yellow is the neutron, and it's charged as 0, while red is your little tiny electron. So now my carbon atom has 6 protons, 1, 2, 3, 4, and there's 2 in the back. That's 6 protons. How many neutrons does it have? 6. And how many electrons? 6. So that means carbon is neutral. That's how it always is. Not always, but for most stable atoms, you will always have the same number of protons and neutrons. They all are equal. They all are equal. Does that make sense? Does that make sense?
Yes.
Excellent. Now let's look at sodium. It has hydrogen protons that are positively charged, and they have 12 neutrons that are neutrally charged. And how many electrons it must have? It must have 11 electrons. So on the exam, I can give you sodium has 11 positive protons, 11 electrons that are negative, and 12 neutrons. What's the atomic number? It's the number of protons only. What is the mass number? It's the number of. Good. Let's look at the nucleus. This is the nucleus, and these are the electron clouds or electron shells around it. Now, we talked about isotopes. Isotopes are variations or varieties of the same element that differ only in the number of the neutrons and therefore in their atomic mass. So their atomic mass is different. So extra neutrons increase the atomic weight. So the atomic weight or the atomic mass. That's why they call the atomic weight the relative atomic mass. So that's what we talked about. That's hydrogen. You see, hydrogen has 1 red hydrogen proton, while deuterium has 1 red hydrogen proton, but it has 1 neutral neutron. And tritium has 2 neutrons. So for you to understand that they must be isotopes, they must have the same number of protons. They cannot change the number of protons. Because if you change the number of protons, you change the actual atom. Then the neutron is what makes them different. So isotopes have the same atomic number, but they have different neutrons or different mass number. Very good, very good. Okay, you see that always to be neutral, you must have the same number of protons and the same number of electrons, the same number of protons and the same number of electrons. Okay. So now, why are isotopes important? Because they ionize and they help us for the radiology, for the diagnostics. Okay, and this is Madame Curie. She was the first female to get a Nobel Prize, and she was the first woman in the world to receive a PhD. So this is just information about, you know, the historical development of radiology, and that isotopes are important for this radiology. Okay, now we talked about how there's a balance between ions and between electrons and protons. Now let's say one cell loses an electron. If it loses an electron, it is now positively charged because it lost the minus. And if it gains an electron, we said electron is negatively charged. So if it gains an electron, that makes it electronegative. So ionization is the transfer of electrons from one atom to another atom. Ionization is the transfer of electrons from one atom to another atom. And the atom that received the electron is known as the atom that received the electron is known as the anion, while the atom that lost the electron is known as the cation. So the atom that received an electron, the anion, is what charge? Is it positively charged or negatively charged? Good. And when the cation lost that electron, is it an example of positively charged or negatively charged? That is, it lost an electron. So that makes it what charge? Positively charged. That's what happens here in the ionization of sodium. So what happens in the ionization of sodium, we have, this is sodium. Sodium has 11 protons, 11 plus, and 11 electrons, 11 minus, and they're both even out. They are both at 0. But sodium gives 1 electron to chloride. So now chloride becomes an ion, and chloride becomes an anion, and sodium becomes a cation. Sodium becomes a cation. Now this is sodium chloride. So the charged sodium ion and the charged sodium ion and chloride ions result like this because sodium lost. You see, it lost 1 electron, so its valence shell went down. So now it's 2. It used to be 3. Look, 1, 2, 3. It lost 1. Now it's 2. While here it gained 1 electron. So now, before chloride had 17 electrons, now chloride has 18 electrons. And sodium had 11, it had 10 electrons. Now it lost 1. It had 11 electrons, and it lost 1. Now it has 10 electrons. So that relationship, that bonding. Okay, let me talk more about this here. Let me stop the sharing for a second. And let me make this big. How do I do that? Okay, so let me explain it like that. This concept is really important. So we're going to talk about the different types of bonding that can happen between the atoms. So here, let's say I had sodium. Let's say I had sodium. And sodium was neutral. Chloride was neutral. Then sodium gave chloride a pair of electrons. What happens now? So the one who lost the electron, Leo says grr. Leo says grr. Leo means losing the electrons. So if you lose the electrons, you are oxidized. So who lost the electrons here? Sodium or chloride? Sodium. So that is oxidized. That is oxidized. So sodium is oxidized. And who gained the electrons? Gained electron reduction. Who gained the electrons? Chloride. So the chloride is reduced. Okay, then the other law says if you are oxidized, then you are the reducing agent. The reducing agent. And if you are reduced, you are the oxidizing agent. So you are the agent of the opposite of that, what happened to you. So if you are oxidized, you are the reducing agent. If you are reduced, You are the oxidizing agent. Make sense? Okay, now let's look at this relationship between sodium and chloride. Sodium gave chloride the pair of electrons. Now they have a bond. What kind of bond is that? It's an ionic bond because they are bonded because of their ions. They are bonded because of their ions. So that's one kind of bonding. That's ionic bonding. Is this the only type of bonding? No. There is hydrogen bonding. So in hydrogen bonding, what happens is it's a bond between oxygen and hydrogen. So in water, you have a bond between oxygen and two hydrogens. So usually the bond is covalent bond. The bond is a covalent bond. So if you have double bond between oxygen and carbon, what happens is you will see that they are shared equally. So oxygen gets 50%, chloride gets 50%. Then that oxygen will get 50% and chloride will get 50%. So that's shared equally. Like let's say my friend and I, we rent a cab to drive Uber. We pay $200, 100 me, 100 him. And then I drive the Uber 12 hours in the night shift, and then my friend drives it 12 hours in the day shift, and we share it equally. That's what happens in covalent bonding. Because oxygen and carbon, they are similar. Now let's look at when oxygen and hydrogen have a bond. Oxygen is so big. So oxygen is now going to drive the cab for 15 hours and give hydrogen like 9 hours. Or maybe drive the car for 20 hours and give hydrogen only 4 hours. Because oxygen is so big, the circles are not going to be equal. Oxygen is going to be so big and hydrogen is so small. Because the electrons are going to be spending more time near the oxygen than the hydrogen. More time near the oxygen than the hydrogen. You understand? So we use that for solvation. Solvation, to dissolve the solutes. So now let's look at what happens. This is very important. Let's say now I have sodium chloride, salt, sodium chloride or calcium chloride, doesn't matter. Sodium chloride. What kind of bond is sodium chloride? Ionic bond. And in the ionic bond, there's somebody oxidized, somebody reduced. There is the oxidizing agent and the reducing agent. Who is oxidized in sodium chloride? Sodium, because it lost the electron. And if sodium is oxidized, what agent is it? Reducing agent. And who's reduced there in the bond? NaCl. Who's reduced in the bond? NaCl. Chloride. And if chloride is reduced, who's the oxidizing agent? Chloride. Excellent. Then when you drop salt in water, water looks like this. OH. Who's more electronegative and who's the most electropositive? Who's mostly electronegative? Oxygen, because it's pulling. I told you that electrons are spending more time around the oxygen than they are with hydrogen. The cab is with the bigger guy more time than it is with the smaller guy. So who is pulling the electrons? Who's more electronegative? Oxygen or hydrogen? Oxygen. And who's more electropositive? Hydrogen. So when you drop sodium chloride in the water that looks like this is mostly electronegative and this is mostly electropositive. Oxygen will say, hey, sodium, you are positive, right? Because it lost an electron. And chloride is negative because it gained an electron. Oxygen will say, hey, I am more electronegative than chloride. Come with me. So it will make a bond with oxygen. So oxygen will break the bond between sodium and chloride and take sodium. And hydrogen will say, chloride, I am more positive than sodium. Come with me. Then chloride is going to bond with hydrogen. And that's how it is. All the sodiums get stolen by oxygen, and all the chloride gets stolen by hydrogen, and that's how we dissolve sodium chloride. Clear? Very important. Let's say now I replace it with calcium chloride. Same thing. Calcium will donate its electrons to chloride, lose electrons, oxidation. So calcium will be oxidized or reduced. Calcium will be oxidized or reduced. Oxidized. Leo says, girl, lose electrons, oxidation. So calcium is oxidized. And if you are oxidized, what kind of agent are you? Reducing agent. So calcium is oxidized, it's the reducing agent. Chloride is reduced, it's the oxidizing agent. Then what happens? The same thing happens when you drop it in water. Calcium is a little more electropositive and chloride is more electronegative. Oxygen will say, hey, have a bond with me instead of chloride. I am more electronegative than the negative chloride that you're with. And hydrogen proton will say, if you are electronegative, then bind with me. And that's how we're going to dissolve sodium chloride. So when you put salt in water and it disappears, you know exactly what happened. That oxygen took the sodium away and hydrogen took the chloride away, and now it disappeared in water. That's how it dissolved. That's how the solvation happens. Solvation happens. So you should be able to explain to me on the exam how does water solvate sodium chloride like that. Clear?
Your exams are going to be written exams?
Yeah, there's some questions that are short answer. Yeah. But I'm going to tell you in the study guide. But for now, you should know how the solvation happens. So we have more electronegative oxygen that will bind with the electropositive sodium, and more electropositive hydrogen that's going to bind more with the chloride. Clear? Okay. That concept was important. How do I get rid of this really big screen? Okay, now we talked about the salts and how we solvate the salts, and now it's time to talk about the electrolytes. So electrolytes are substances that ionize. So when you break NaCl, and now it's positive calcium, negative chloride, or you have Na plus Cl minus or Ca plus Cl minus, that's an electrolyte. So if you have pure water and you run an electric current through the pure water, you're not going to get electrocuted if you dip your finger there. But if you put electrolytes, then these electrolytes are going to be able to mediate the transfer of electricity to your fingertips. So electrolytes are substances that ionize in water. Ionize meaning one becomes positively charged and the other one becomes negatively charged. So the chemical reactivity, osmotic effect, and electrical excitability of the nerve and muscle cells. So electrolytes balance is one of the most important considerations in patient care. So we need to avoid acidosis and alkalosis. Free radicals, we talked about the SOD. We have talked about the superoxide dismutase. So these reactive oxidative species, we talked about the other day how they are used to destroy bacteria. So your macrophage has a lot of reactive oxidative species, and the reactive oxidative species destroy the integrity of the virus, destroy the integrity of the bacteria, destroy the structural integrity of the parasites and the fungi, and it does that with free radicals. But the problem is the people who consume alcohol. Alcohol gets metabolized in the body into acetaldehyde, and it increases the reactive oxidative species. And that reactive oxidative species is now going to start damaging the liver. And that's how we have alcoholic liver disease. So one of the main dangerous free radicals you're concerned with is superoxide ion. So O2, what is O2? Oxygen. That's very nice to have, O2. But what about O2 minus? That's superoxide. So O2 minus, that is superoxide. Now superoxide, there is an enzyme that helps us. It mutes this oxygen minus. It mutes the oxygen minus. Oh, very good. Now I can use that. Okay, so we have O2 minus. We don't want it. That's a superoxide. Superoxide. So superoxide, there is an enzyme known as SOD, S-O-D, superoxide dismutase. So the superoxide dismutase is going to change O2 minus into H2O. It's going to change it into, into oxygen, O2, plus hydrogen peroxide, H2O2, hydrogen peroxide. Okay, so you must remember that, the SOD, the superoxide dismutase. It takes the dangerous superoxide O minus and converts it into useful, very good oxygen that we can use for oxidative phosphorylation and for aerobic respiration. And we use H2O2, and that is hydrogen peroxide. It's also useful. It's also useful for us.
You're saying this superoxide, it becomes
oxygen, and
it splits.
Yeah, it splits into oxygen. It is converted into oxygen and hydrogen peroxide with the help of SOD, superoxide dismutase. So this is a free radical. So the superoxide is a free radical. Is a free radical. And the free radical is going to be converted into, is a dangerous. The free radical is dangerous, is negative, is stressful. It gets muted into oxygen and hydrogen peroxide through the actions of superoxide dismutase. This is an enzyme. Superoxide dismutase is an enzyme. Superoxide dismutase is an enzyme. So that enzyme converts for us superoxide into oxygen and hydrogen peroxide. Okay? Yes.
Okay. So the superoxide is the H.
Okay. Okay. So molecule is a particle composed of two or more atoms united together by a chemical bond. And the compound is a molecule that is composed of two or more different elements. And it can be represented by a molecular formula, which identifies constituent elements and how many atoms of each are present. So what are isomers? Isomers have the same number of molecules. Isomers have the same number of molecules. Then what is the only difference? The difference is that even though they have the same number of molecules, they are structured differently. Now let's look at ethanol here. You guys see ethanol in the screen? It has two carbon atoms. Two carbon atoms. How many oxygen? One oxygen, one oxygen. How many hydrogen? 1, 2, 3, 1, 2, 3, 6 for both. But are they arranged the same way? Here, oxygen is in the middle. Here, oxygen is in the far to the right. So that shows you the molecular formula is the same. Both of them have two carbons. Both of them have 6 hydrogen. And both of them have 1 oxygen. But the structure is different. For ethyl ether, oxygen is right in the middle, like here. While in ethanol, oxygen is far to the right. You get it? Okay. The molecular weight of a compound is the sum of the atomic weights of its atoms. So if I want to know the molecular weight of glucose, I go to my periodic table. Carbon, what's the AMUs for carbon? What is the molecular weight for carbon? What is the molecular weight for carbon? 12. You see, that's the atomic number. That's the number of proton. Molecular weight is 12. Very good. Molecular weight is 12. They have, so now if I want to find out how much glucose is, so I go, this is a typo, it's not 66 carbons, it's 6 carbons, because glucose has 6 carbons, 12 hydrogen, 6 oxygen. If I want to know the total molecular weight for glucose, I multiply 6 by its atomic mass unit. So now I know that carbon is 12. So I multiply 6 by 12. Hydrogen is 1. Hydrogen is 1. So 12 times 1, and oxygen is 16. Oxygen is 16 right here. So then I multiply and then I add them up all together, and that will give me the molecular weight. Any questions?
So wait, to get the molecular weight,
you add the molecular mass for each one. For each one. That's like you want to know how much your salad weighs. It has 2 tomatoes, and it has 2 onions, and it has 2 tomatoes, 2 onions, and 2 oranges. So you add, you multiply 2 orange, each one weighs how much? 2 tomatoes, each one weighs how much? And each, so that's what we're doing here. There's 6 carbons, 12 hydrogens, and 6 oxygens, right?
right?
So now I go to my periodic table. Carbon is how much? 12, right? The mass for carbon is 12. Carbon weighs 12. So I multiply how many carbons by 12? There is 6 carbons, so 6 times 12.
So the mass of the element multiplied by the amount.
Yeah, by how many that we have.
Oh, okay.
Yeah, and then we add them up all together. That comes up to the molecular mass of all of them, because all of them share together. Okay? So if there are 3 men in one room and 2 women, the mass of the people is each man worth how much, each woman weighs how much, and they add them up together. Okay? Now let's look at here. This is an example of a hydrogen that's a single covalent bond. So we talked about the types of bonds. There's an ionic bond, and these are bonds that there's an attraction of a cation to an anion. And an example would be sodium and chloride ions, and they bond to form sodium chloride. So that's relatively easily broken by something that's more attractive, such as water. We talked about how water breaks the ionic bond. Then the covalent bonds, we have single bonds. We have single covalent bond. That is a nuclei that is shared between one pair of electrons. Double covalent bond is a nuclei that is shared between 2 pairs of electrons. So if electrons are shared equally, it's a nonpolar covalent bond, meaning that carbon atoms are bonding together. So if electrons shared unequally, it's a polar covalent bond. That's what happened here between oxygen and hydrogen. So the electrons are spending more time around the oxygen than they are around the hydrogen. That's polar. So they are polarized to one side more than the other. So that's an example of a polar covalent bond, because it's a covalent bond. We share electrons, but even though we're sharing them, one is bigger than the other. So that's what happens in water. That's a polar, when they are unequal. Okay? However, if they're shared equally, like between carbon and oxygen, that's an example of both are equal size. Both are equal size. Okay? So that's an example of 2 hydrogen atoms attached together. Okay? And that's a double covalent bond. So 2 electrons and 2 electrons attached together. And here the oxygen atoms, 2 electrons, and 2 electrons are shared. You see? Both of them are shared equally. So that's double covalent bond. So which one is more stable? A single covalent bond, a double covalent bond, or a triple covalent bond? Who's the most stable? 2 bonds, 3 bonds, or 1 bond? Stable. Which bond is the most stable? With triple bond. You are most stable if you have triple bonds. Okay? If you tie something up with 3 strings compared to 2 strings, compared to 1, which one is more stable? 3. So the more number of bonds, the more structural stability that we're going to have. Okay, so that shows you nonpolar versus polar covalent bond. So you see the polar covalent bond, the nonpolar covalent bond is shared equally between both carbon ions. While the polar covalent bond, even though it's a covalent bond, but the electrons are more oxygen is more electronegative, and the bubble around oxygen is bigger than around hydrogen, indicating that electrons are what? Here, in this relationship here, you see my mouse? This relationship here between oxygen, that is red, and hydrogen, that's plus. That shows you that electrons are spending more time around the oxygen than around the little hydrogen, because oxygen is pulling them away. Oxygen is more electronegative. And hydrogen is more electropositive. So hydrogen bond is a weak attraction between a slightly positive hydrogen atom in one molecule and a slightly negative oxygen or nitrogen atom in another atom. So relatively weak bonds, but very important to physiology. So the water molecules are attracted to each other by hydrogen bonds. They are attracted to each other by hydrogen bonds. And the large molecules of the DNA, like for example, DNA, there's a double helix and both are tied together, double helix and both are tied together due to hydrogen bonding. So that shows you the usefulness of hydrogen bonding. Why do we have hydrogen bonding? To solvate, so water can dissolve. And also we use hydrogen bonding so we can have larger molecules like DNA and proteins bonding together. Okay, so now we're talking about the bonding to prepare ourselves to learn about the structure of these macromolecules. You know, we said today our objective, we got to know what a protein look like. We got to be able to tell what amino acid that builds up a protein looks like. What is a fatty acid triglycerol? What is a fatty acid, free fatty acid, triacyl glycerol looks like, and what does the nucleotide look like? You got to be able to tell that's a nucleotide, that's an amino acid, that's a sugar. You got to be able to tell that. So we are learning the bonds. So later on, when we look at this structure, you can tell that's a covalent bond, that's a hydrogen bond, that's an ionic bond, because all of these bonds come to in the structure of all of these macromolecules. So we are preparing to go now and learn about. So now that's what I was talking about earlier. You see, oxygen is most electronegative. So that's how the water look like before you add anything. So the water is like H2. There's two H's and one O. Hydrogen is more electropositive. Oxygen is more electronegative. Hydrogen is more electropositive. Oxygen is more electronegative. But they connect where the electropositive is touching the electronegative. You see, the electropositive is touching the electronegative. Electropositive touching the electronegative.
That's why it stays together.
Yeah, that's how it stays together. But when you drop sodium chloride in the middle, then they're not going to bind to each other. They're going to go and bind the sodium chloride, break it, and now the oxygen will bind to the sodium and the hydrogen will bind to the chloride. There is another type of bonding known as the van der Waals forces. They are also weak and brief interactions between the neutral atoms. And these are fluctuations in electron density within an atom, and that creates the polarity for a moment and attracts the adjacent atom for a very short time. So only 1% as strong as the covalent bond, but play important role in physiology. Now, mixtures. What are mixtures? Mixtures are, so before we go into mixtures, let's take a break. Yeah, let's take a break and come back.